and welcome to the National Research Collaborative Meeting 2020. Thank you for joining us. We're excited to present our third webinar, Efficient Clinical Trials, an introduction to pragmatic trial design with discussion of some of the key considerations surrounding designing trials. Now, I'm so pleased to introduce our chair for the evening. Professor Marion Campbell is Vice Principal for the University of Aberdeen and Professor of Health Services Research in the Health Services Research Unit. She's a medical statistician and clinical trialist, and her main research interests are in the design, conduct, and analysis of clinical trials, especially complex trials and the design and conduct of surgical and device trials. Welcome, Professor Campbell.
Well, thanks very much. I'm absolutely delighted to be here tonight. What an exciting lineup and an exciting set of speakers to speak with you tonight. We are going across the pond to Canada, to Ottawa. We're picking up from McMaster and we're coming to Aberdeen as well. So I'm just going to let you know who the speakers are, and then without further ado, we'll get into the exciting science. But before I introduce our speakers, I do want to encourage you to ask questions. This is going to be a chat, an interaction, and it's a fantastic opportunity to ask all those questions you want to find the answers to. So it's as good as your interaction. Otherwise, I'm going to hog all the questions, and that would be very dull. As I say, we've got 4 speakers tonight, and I'll just let you know who they are. So we've got Mark Clemens, we've got Gregory Pond, we've got Sharon McKee, and we've got Sean Treweek. And so each of them are going to have 10 minutes or so to give a presentation. But first of all, we're going to turn to the first speaker, who's Professor Mark Clemens. And now let me introduce you to him. So Mark is a clinical investigator at the Ottawa Health Research Institute and a staff medical oncologist at the Ottawa Hospital Cancer Centre and a professor of medicine at the University of Ottawa. And he's involved in a large number of studies for patients with early stage, locally advanced and metastatic breast cancer. And he created the Rethinking Clinical Trials program, the REACT program, which places the patient and their family at the core of designing and implementing trials that actually direct and improve patient care. So Mark is going to talk to us today. The title of his talk is An Introduction to Pragmatic Trials and Integrated Oral Consent. So I'm going to hand over to you, Mark. Looking forward to it.
Lovely. Thank you very much for the invitation to speak to an audience who's back at home for me. So my mandate is essentially a trial for every patient and a patient for every trial. And I'm going to talk about the real-world experiences of real-world trials. My talk will be followed by Greg Pond, who's going to talk more about statistics, and then Sharon McGree, who's going to talk about a real-world practical trial that integrated oral consent in its methodology as a working example of how these kind of trials should be done more and how all of us can be involved. I'm going to talk about what the state of cancer trials is at the moment. As an oncologist, I'm going to talk about the REACT program, that's the Rethinking Clinical Trials program that we've been running here in Canada for about 6 years now. And I'm also going to talk about integrated oral consent for standard of care options, which is particularly important for discussing surgical and device trials where many different options are considered standard. So we're calling today from Ontario, Canada's largest population by province, and we can see from this graph that despite the fact that the incidence of cancer has risen over recent years, the proportion of patients being enrolled in clinical trials is now less than 3% of adults, and many of these are for phase 1 and phase 2 trials where the benefits for the individual patient are often marginal at best. Why is accrual so low? And you'll see many of these slides presented where it talks about physician engagement, the efforts of the consent process. My 2 spiraling yellow arms are to show how sometimes contracts and REB can take a significant length of time. There's cost, there's superfluous data collection. Some people say patient engagement is one of the challenges. And I disagree. I think the elephant in the room is the biggest barrier for clinical trial accrual is physician engagement. If a physician does not approach the patient about the study, they will not be enrolled. COVID is affecting all of us across the world, and this paper came out in JAMA last week. And essentially what it's saying is patients since the arrival of COVID, irrespective of their age, sex, income, are less and less interested in being enrolled in clinical trials at the moment. And the graph on the right-hand side shows that the biggest concern for patients at the moment is fear of exposure to COVID. And I'm sure you've all found that your clinics are becoming more and more virtual. So if we're not seeing the patients in person, we need to find a better way of doing our trials. So the paradox as I see it is we have more and more cancer patients. We know more and more about cancer. We have more treatments and our healthcare budgets are growing almost exponentially. However, we have little guidance in many areas of care that actually impact on how real patients do in the real world, how treatments affect their quality of life. A personal interest of mine is healthcare system sustainability. How do we avoid burnout? And also, we have more and more reasonable treatment options, be they cancer therapy, a device, a surgery, to offer patients. A simple example that I like to use is the fact that if I get the results of a midstream urine, and all of us get these results most days, it shows us E. coli. You have a choice of antibiotics to give, but which is the best? And we almost never know the answer. And this is what we term the eccentricities of physician decision-making processes. The physicians tend to use the one they're used to using, and sometimes they'll base their decision on what's funded. Rarely is it based on actual evidence comparing these different antibiotics. What are pragmatic trials? You're going to hear more about them as the evening goes on. Explanatory trials are where we use a well-defined and controlled setting. These trials traditionally select for patients who really don't, on the whole, represent the broader population of patients we treat. They're younger, they're richer, they have less comorbidities, and they take less other medications. Whereas pragmatic trials are where you're testing an effectiveness of intervention in broad, routine clinical practice. So the REACT program has been going, as I said, for about 6 years now. And these are the elements of the program. But for tonight's discussion, it's really around performing surveys and systematic reviews to demonstrate equipoise, and then the integrated oral consent model. So before planning any study, we will ask patients and healthcare providers, what are you doing today? And in particular, we will ask patients what treatment you are getting. And also we ask in our survey, what trial endpoints are the most important to you? If we're going to do trials, what do you want the endpoint to show? Because often we find what healthcare providers in all their wisdom want is not always what patients want. The other advantage of doing these studies is the survey is it would get publications for our trainees and more data for applying for grants. The traditional consent process, this is supposed to be me talking to the patient. I leave the room, then the research staff come and talk to the patient, often with some delay. The patient is then faced with a very long consent form that then allows various blood tests and scans to be performed. And then the patient's back with me to talk about the trial again. It is not surprising that every step leads to loss of patient interest in trials and also significant cost for actually doing studies. So the integrated consent model is what we've been developing based on UK data. And essentially, it's me talking to the patient, a one-page consent template, oral consent, and randomization there and then in the clinic. So, for example, one of the trials we did, and Sharon will talk about this more in a few minutes' time, there's a drug that we give as an injection to boost the white cells during chemotherapy. We can give 5, 7, or 10 days of injections. All of them are reasonable. All of them are a standard of care. We want to do a trial comparing them. Are you interested? That is integrated consent. I then will dictate that these injections are given for reduction of risk of infections, 5, 7 days, or 10 days are standard. I've given the patient the consent template, and randomization has been done there and then. This is very different than the 20-page consent model that is often used in other types of trials. And I think many of you in the audience will realize this is exactly the sort of thing that we can do in our clinic and at the bedside. With the app that we use, we have very few eligibility criteria because we want this population to represent real patients that we see. So there's a brief checklist, and once we've entered the patient's name and hospital number, it will email us in the clinic when the patient's due back to capture data. So it's very practical for what we actually do in real world practice. The other thing is we do not bring patients back for trial defined visits. Visits are all done as per standard of care. We've now enrolled close to 3,000 patients into these REACT trials. With a surgical audience, I put those to the left-hand side, but we've done supportive care, adjuvant trials, device trials, imaging studies, and palliative care studies. The surgical trial that we've done, the first one, was looking at 2 different types of mesh for breast reconstruction. And we have a large study running at the moment of preparing patients for colorectal studies. What is the best form of preparation of the patient for the surgery? Is it mechanical bowel preparation? Is it oral antibiotics? Simple, practical questions. So what are my conclusions? I would say to everybody, ask questions in the clinic. Dogma is just that, dogma. Ask yourself, why do I do such and such for this intervention? Why do my colleagues do something else? Who's right? Who's wrong? You're probably all right. REACT is not perfect. We still need phase 1, 2, and 3 trials. With any clinical trial, the more you ask healthcare providers to do, the less they will do. So asking for more gets you less. This is important for your methods and your trial design. We use integrated oral consent for comparing standard of care options. This is not oral consent for phase 1, 2 trials. This is where we have multiple standards of care. COVID-19 has made all of our lives challenging, but I'd like to say it's also potentially exciting because we have to design our trials now based around the fact that patients are going to be coming less and less to the clinic, and we need to be able to consent and collect data by telephone and email. With that, that's my talk completed, and we'll go back to Marion, who will introduce the next speaker.
Excellent. That's already given us a lot of food for thought. The idea of being able to randomize in the clinic setting is fantastic, and I'm sure that's going to generate a lot of questions as we go through the session. So let's move on. I thought we'll do all the talks together because they're interrelated, and it'll be important. Some talks will raise other questions for us. So the second speaker tonight is Dr. Gregory Pond, and he is a biostatistician specializing in cancer research. He's an associate professor in the Department of Oncology at McMaster University in Hamilton, Ontario, and a member of the Ontario Clinical Oncology Group. He's also an investigator at the Escarpment Cancer Research Institute in affiliation with McMaster. And his main research interests is the statistical analysis of cancer clinical trials with the aim of translating scientific discoveries into the clinic. So Greg's talk tonight the title is Statistical Considerations, Why Endpoints and Sample Size Are So Important. So it's over to you, Greg.
Thank you. So thank you, Marion, for the introduction. So the title of my talk, The Importance of Endpoints and Sample Size Effects. I think the importance, I think most people here would be readily understand the importance of them. So I changed it a little bit and want to talk about how different endpoints for a very standard or potentially a simple clinical trial scenario can greatly affect the sample size and the interpretation. So I'm going to just briefly go through my disclosures. And what I'm going to do and talk for the next 10 minutes is just present you with a relatively straightforward scenario, and then, as I said, discuss how the different outcomes and the sample size implications, as well as the interpretations of the results, and how they can be make different inferences based on what outcomes you end up choosing. So the scenario that I chose is an actual trial that I was working on a little while ago. And so we're looking at Gleason grade 2, which is formerly known as Gleason 3+4 prostate cancer patients. This is a population of generally elderly men. There's often some mild comorbidities associated with these men. And at least in Canada, from my colleagues, what I've been told, the standard of care is one of two options. The first one is an active surveillance. Basically, there's no real therapeutic benefit at the time. They just follow them up and will have potentially repeat biopsies, follow their PSA. Again, because the men are generally elderly in this population, a lot of these patients will die not of prostate cancer, so active surveillance is believed to be a good potential option for these patients. The other standard of care option is radical radiotherapy or radical prostatectomy. Now, there's a definite therapeutic benefit, but there's a lot of side effects, greatly affects the patient quality of life. It's kind of on the opposite. So while there's definitely an improvement in terms of the prostate cancer and the efficacy related to the prostate cancer, it affects the other avenues of the patient in terms of their quality of life. So there's these two options that we presently have. Now, in this particular scenario, there's a new treatment called focal ablation therapy, which has been proposed for investigation. Now, if this was successful, the lead investigators wanted to apply this for regulatory approval, whether through Health Canada in Canada, the FDA in the US. And so they used the FDA guidance in looking to see at what they needed for regulatory approval. And they're basically coming to the conclusion that you need to demonstrate clinical benefit. That's what the FDA guidances say. And so the question really comes out to, how do you define clinical benefit, particularly in this population? So the easiest, and I shouldn't say the easiest, the most objective measure in terms of clinical benefit is overall survival. There's a definite defined endpoint. Everybody eventually will die, so there's a defined endpoint for every single patient. But again, in this particular population, the prostate cancer, because it doesn't necessarily kill a lot of the patients, they will die of other things. So the 10-year overall survival, even in the active surveillance arm, and I've given one particular reference, is very high, potentially 92%. So just to see a small improvement over the active surveillance arm, say from 92% to 94%, you could have a very large study of over 16,000 patients, which are required, and it can take a very long time. Again, 5 years plus follow-up plus time to prepare and develop the study. This could be a 7, 8, 9-year study. And for the study investigators, they needed something much quicker than this because, again, they're looking to get FDA and Health Canada approval within a matter of a couple of years. So this was something that, although is a good long-term option, is really not beneficial for what the investigators were looking for at this time. Now, there's other similar types of endpoints, survival type endpoints, that were also bandied about by the investigators at different times. Potential options that you could look at. So prostate cancer specific mortality. Now, this does have fewer events than overall mortality, but is actually specific because, again, overall survival, a lot of these patients do die of other things. Prostate cancer specific, we know that it's related directly to the prostate cancer. So if the treatment was effective, it would greatly reduce the prostate cancer mortality. Of course, some of the difficulties are, how do you actually confirm the cause of death in most of the men? Again, if with many of these elderly men, when they die, they do not undergo an autopsy, and the cause of death as listed on the death certificate is sometimes not thoroughly thought through, I would say, and just, they have cancer, they write it down, well, they died of cancer or something like that. So it's not necessarily prostate cancer specific mortality. It's kind of a difficult thing to measure. Another possible outcome that people we talked about was time to progression. Well, in this case, if you have a non-prostate cancer death it's a competing risk. But again, this relies on having a good cause of death associated with the death certificate, which doesn't always happen. In addition to that, in this particular population, there's some questions on how do you determine progressive disease. For many of these men, after they undergo biopsy once, they tend not to want to go under biopsy a second time, so you can't use a biopsy as a measure of progressive disease. Some of the other outcomes that they use presently for progressive disease may not be absolutely perfectly objective, like PSA progression or something along those lines. So the determination of what is progression may also be very difficult. So again, these are all the different considerations that you have to consider. Now, going back to the actual scenario that we were talking about, what are some of the inferences that you can get when you start thinking about what are you comparing, and what's the focal ablation versus active surveillance or radical treatment? Well, theoretically, focal ablation should be better than active surveillance because you're giving some sort of treatment, regardless of the endpoint. So just having some measure of improvement in terms of efficacy may not be enough, because again, a lot of these patients don't go on radical treatment right now because of the side effects. So part of the question that you have to answer is the quality of life of these patients appropriate? But then that goes against the radical prostatectomy arm or radical radiotherapy arm, when focal ablation is a less harsh treatment compared to these radical treatments. So you should have improved quality of life. Again, if you're looking at these long-term outcomes, thinking about the non-prostate cancer deaths, that happens a lot in these particular patients. So how representative is overall survival or some of these other markers of efficacy, given that many of these patients die of other things? And as I mentioned, when you compare the ablation versus radical treatment, if you're looking at the quality of life, you're expecting to see an efficacy or a quality of life improvement or a toxicity improvement. And so what you really want to look at, if you're looking at these efficacy outcomes, is a non-inferiority question as opposed to a superiority question if you're comparing focal ablation with active surveillance. So again, depending on what the comparator is in this particular setting, may change the design from a superiority to a non-inferiority question or vice versa. Now, just to give you an idea, another possible thing that we talked about was time to more aggressive treatment, which was kind of a short-term, potentially surrogate outcome compared to overall survival or time to progression, that sort of thing. And so the thought was that a delay to more aggressive treatment gives an improvement in the patient quality of life. Again, just in terms of how the outcome may change the sample size, if we use 7-year treatment-free survival of 42%, which I found quoted in a different publication, if you had it, we're looking for an improvement of about 10% from 42% to 52%, your sample size would change to about 1,500 patients, again, depending on the length of follow-up that you have. So you can see how a small change in your outcome may have a big change in your sample size calculations. But then again, going into the inferences here, by definition, focal ablation will be better than any of the radical treatments because all the radical treatments have a more aggressive treatment on day 0. That's by definition. So they're automatically getting it. So you can't really have a comparison in that sense. So you can only compare focal ablation with active surveillance. And so the question is it delaying time to radical treatment or time to more aggressive treatment compared to active surveillance? But you have to really find a very strong benefit in this particular case because you are giving some sort of treatment. And by, theoretically, we should expect some treatment is going to have an increased cost. You're going to have some side effects, even if they're small or minor, and compared to an active surveillance. So you need to see a very strong superior difference. Again, one of the things in terms of a treatment choice, one of the problems with this is the patients actually have a choice whether or not they're going to undergo this future treatment. And a lot of the patients will opt for the treatment just because they have a fear of the cancer returning or the progressive disease. Some patients will just refuse the treatment because they don't like the side effects. So there's other options that are affecting the outcome, not just the efficacy of the treatment itself. The patient has a great decision to make on, or a great influence on whether or not the outcome is going to occur or not. So it may not necessarily be the best outcome. Again, so another possibility that we looked at is, let's just look at the toxicity. How does the focal ablation compare to the other treatments in terms of the toxicity rates? So I gave you the toxicity rates here, depending on if it's grade 1, grade 2, or grade 3. But now you have to start thinking about, if you're planning a trial based on the toxicity rates, which one is the important one? Is it any toxicity, grade 1 or above? Or does it have to be a grade 3 toxicity? Are you only focused on GU toxicities, or do you need to have GU or GI toxicities? You have to get all these questions and decide what is actually clinically important to the patient in terms of the outcomes. But again, just to give you an idea, how does this change the outcomes? If you were looking at, say, grade 2 and above GU toxicities, the rate was 54%. So if you want to reduce it to 44%, you only need about 800 patients. Or if you want to look at the grade 3 toxicities, your sample size goes down to about 1,374 patients, based on reducing the toxicity rate from 9% down to 5%. Now, alternatively, if you're only looking at toxicity, perhaps you don't need a randomized trial, you only need a single-arm trial. Again, this could vastly affect the sample size that you're looking at. Using the same estimates in terms of the toxicity rates, you may only need 200 patients or 300 patients, depending on the toxicity rate that you're actually interested in. So again, you can have vast differences in terms of what this sample size would be, depending on what you're actually looking at. Now, toxicity isn't the only measure of quality of life or how things are going. So quality of life, you have a number of choices that you could also alternatively use, and not just a straight toxicity rate. But again, you have to decide what's the quality of life measure that I use. Do you use the SF-36, EPIC? There's all kinds of different quality of life questionnaire surveys that are out there. And they all have actually a bunch of subsections, which maybe it's one particular subsection that you're interested in, not necessarily the entire global quality of life score. In addition, you have to look at, are you interested in the absolute score, the change in the score? Are you looking at adjusted scores based on other factors that may occur? Are you looking at a time component? And what I mean by that is, what's the time after treatment that's really important? Is it at 3 months after treatment? Is it 6 months? Is it 2 years? Or is it across the entire spectrum of time after treatment that you're really interested in? Again, depending on the score that you can use, you may have vast differences. So for instance, there's one paper here which showed that the EPIC urinary function had a 15-point change compared to for prostatectomy, whereas active surveillance basically had no change, as you would kind of expect. But if you looked at those particular numbers, the sample size that you would need for using this particular score as a measure of quality of life, you'd only need about 30 patients. So again, vastly different than the 16,000 that you would need for an overall survival. But again, you have to consider what are the outcomes that what are the inferences you're going to have at the end of the result, at the end of your study. Again, active surveillance should theoretically have 0% toxicity, because you're not actively treating the patient at that particular, you're just following the patient over time. It should also have no change to the quality of life. So by definition, active surveillance should be better than focal ablation. So if you wanted to use a quality of life measure, maybe you really only can compare to the radical treatments, radical prostatectomy. By definition, or theoretically, focal ablation should have a better quality of life and a better treatment than radical treatment. But again, if you think back, well, the active surveillance with 0% toxicity and no change in quality of life is going to have better outcomes compared to the radical treatment. So fewer treatments will just give you less adverse events. And therefore, it doesn't necessarily mean that it's better. It's giving you a different outcome. And again, as I said, what's really important? Is it the quality of life now, or is it the quality of life 5 years from now or 10 years from now? Maybe when you do need treatment, if you had active surveillance. So these are all questions that you would have to consider. So ultimately, the sample size, whatever trial design that you end up using, single arm, randomized two-arm trial, superiority design, non-inferiority design. They can vary wildly in the same scenario, in the same setting, depending on the outcomes and the objectives that you're really interested in. There is no one-size-fits-all approach. And really the best thing that everybody should do, all investigators, is really think through all the possible results and all the interpretations or inferences, not necessarily from your point of view, but from the point of view of an optimist and from the point of view of a pessimist, in terms of how it would be interpreted and what can be the results as at the end of the study. Once you start really thinking through all these alternatives, then you can come up with what is the best design and the best outcome for my particular question that I'm going to ask. Thank you, and I'm going to pass it back to Dr. Campbell at this point for the next speaker.
Super, that's really interesting. The trade-off between which outcome you choose and what the sample size is, it's one of the big issues that we have to think about when we're designing our trials, and I think that'll raise quite a number of questions when we come to the question bit at the end. So let's move on then. So next up is Dr. Sharon McGee, and she's a medical oncology fellow and assistant professor of medicine at the University of Ottawa. She's a clinician investigator at the Ottawa Hospital Research Institute and a principal investigator for the REACT trial. Her specialist interests are breast cancer and oncofertility and the management of side effects and toxicities related to cancer treatment. So Sharon is going to speak tonight, and the title of her talk is the REACT-RET trial, a practical example of a pragmatic trial. So over to you, Sharon.
Thank you so much. Good evening, everyone. Hopefully you can all hear me. So my name is Sharon McGee, and I'm a medical oncologist at the Ottawa Hospital Cancer Centre in Canada. And I'd like to talk to you this evening to give a practical example of what a pragmatic clinical trial looks like. This is a trial that I am principal investigator for, and it's looking at the timing of radiation and endocrine therapy in patients with early stage breast cancer. So again, just hoping to give you a brief overview and a quick presentation today to really highlight some of the key steps in developing a pragmatic clinical trial to give you an idea of what's involved. And also then finishing with a few points on why I and we think the REACT pragmatic clinical trial program works, and why you should certainly, consider it establishing a similar setup in your own institution. So the first step in designing a pragmatic clinical trial is to identify your research question. And as Mark alluded to before, these questions are really all around you. If you take a look around in your outpatient clinics or in the theater, there are several things that we do on a day-to-day basis that we really don't have a lot of evidence to guide us on. As an oncologist, and particularly focusing on breast cancer, which is one of the most common or the most common cancer diagnosed in women, the majority of my practice will present with early stage disease, and that is usually a hormone receptive disease, or basically breast cancer that's growing in response to estrogen and or progesterone. The main part of treatment for these patients is going to be endocrine therapy, so things like tamoxifen as well as radiation. And although we've been using these treatments really for several years now, we actually don't have a definitive answer as to what the optimal timing of these therapies is. And as such, practice in the clinic is highly variable, with some patients starting the treatments alongside one another, and other patients maybe completing radiation first and then going on to start their endocrine therapy. So the question arose as to, is there an optimal sequence or timing for these therapies? Should they be given concurrently, or should it be something that's done sequentially? The second step that we follow then is to demonstrate clinical equipos. And that essentially means that you're showing or demonstrating that your clinical question, is something that is as yet unanswered by the available data. And this is a really important part in the process because it allows you to review the data to help drive the future design of your clinical trial. And it's also a step towards starting to generate research papers and publications, which we all know is important in academic medicine. Towards our research study and this clinical trial, we conducted and published a systematic review on the available data regarding the timing of radiation and endocrine therapy in early stage breast cancer. From this, we were unable to answer whether there was an optimal treatment sequence, and it was also unclear if there was any impact on the efficacy or toxicity of treatments with the timing. We also conducted and published a survey of Canadian physicians that likewise demonstrated that there was highly variable practice with respect to the timing of these treatments. And we identified that some of the concerns that physicians had regarding the timing of these therapies were related to the potential for toxicity with endocrine therapy or radiation, impacts with compliance to endocrine therapy, as well as potential impacts on the effectiveness or efficacy of radiotherapy. In this survey, we were also able to determine that physicians were very much interested in conducting or designing a pragmatic clinical trial to try and answer this question. And we were also able to identify from them what key or meaningful endpoints would be for them. Step 3 in the process is then, to design your trial. And the big thing I will always say here, and which we kind of try to adhere to, as Mark has said, is to keep it simple. And keys to success that we have found in this regard from various pragmatic clinical studies that we've developed here in Ottawa have been, number one, to have a defined clinical question and hypothesis. It's also important to make sure that your patient population is focused. The endpoints you choose for your study are also very important, and they should be simple and clinically relevant. And they should also have the ability to give you results on both a short and long-term basis. In oncology, we're often driven by endpoints like overall survival and disease-free survival. However, these can take many, many years for final results to come. So if you do want to have some results before potentially you're retiring, it's important to have a more shorter term endpoint. It's also critical as part of your team that you have someone with, a sound background in statistics, such as Dr. Pond, who really is essential in guiding us, making sure that our statistical plans are sound, and that whatever our endpoint is that we're choosing, we have the most appropriate sample size. Now, this can always change as the trial is being conducted, but these are key points at the outset when you're designing your study. You should always also make sure that the process for data collection is simple. The simpler it is for the patient, for the physician, for your clinical research assistants, the more successful your study is going to be. And having lots of people review the study protocol is very important too, because it makes sure that the clinicians are on board and they feel that the clinical question is relevant and meaningful. The statistics is sound. Your CRAs or research administrative staff feel that this is something that they can feasibly do within the clinic. And it's also helpful to know your ethics board within your institution, so to help make sure that you don't get the bottleneck of getting your trials through approval and open. In the case of our study, so REACT-RET, which was looking at the timing of endocrine and radiation therapy in early stage breast cancer, our central hypothesis here was that concurrent endocrine and radiation therapy would be non-inferior to sequential treatment. The primary outcome we chose was endocrine therapy toxicity. And this essentially came from the survey that we conducted of Canadian physicians, as this was one of their key concerns and one of the endpoints that they identified as being most meaningful to them. It's also a more shorter term endpoint. That readout can be available within the course of a few months to a year. And there also exists a validated and established quality of life assessment score that can be used to assess this. Additional secondary outcomes that we chose, again guided by the results from our survey of physicians, were related to radiation toxicity, endocrine therapy compliance, overall quality of life, as well as cost effectiveness. Step 4 in the approach is then, once your trial has been designed and hopefully gotten through approval, that you're able to approach patients. And this is really the fun part of the process. Keys to success here are making sure that you have physicians who are aware of your study and who are engaged and open to approaching patients. And what really helps here is that, again, this factor of making things simple for everybody. Whilst we did say that you need a defined population, it's important that that's a relatively large population so that you have a good chance of accruing patients quickly to the study. As Mark mentioned, really central to the success of these pragmatic clinical trials is this verbal consent model, where physicians can approach patients right there in the clinic, explain the study, obtain verbal consent, quickly randomize them using a web-based tool to have an answer there and then as to which arm they're going to be participating in, and just simply documenting that in the medical record. For us, essentially, it's in their clinic note for the day so that they can begin, participating in the trial. Keeping it simple and easy for everyone is really a key to success throughout this whole process. So for this study, again, we had a defined but broad population with early stage breast cancer patients having hormone responsive disease who were planned to receive treatment with endocrine and radiation. They had to be over the age of 18 and able to give verbal consent. There were really very few exclusion criteria, so this is quite a large population that we see in the clinic. Those eligible patients were able to be approached by either their medical or radiation oncologist, as I said, obtaining sort of verbal consent there and then, as well as randomizing there and then within the clinic so that patients can begin the process of collecting data for the endpoints, which for us, the primary one being the impact of the timing of treatment on toxicities with respect to endocrine therapy particularly. This is just a recent slide showing the patient accrual to this trial. So our sample size here was 218. The study opened in September of last year. The latest figures from this month have shown, actually it's an update as of today, rather than 214, we have 215 out of the goal of 218 patients recruited. So this really represents one of our most successful and fastest accruing pragmatic clinical trials, and primarily because it hits many of those factors that we reviewed before in terms of being a large but defined patient population, physicians who are engaged both within our medical oncology department as well as our radiation oncology department. And I'd also like to point out that, this accrual, this excellent accrual really continued right throughout the COVID pandemic, which we and you have seen has really changed clinical practice with the rapid switch to virtual care. And we were able to continue offering and enrolling patients in clinical trials like this throughout that time, just by making some obviously different changes and compromises. The biggest impact for us was really the ability to assess radiation toxicity, which is something that's performed by the physician during a physical exam. However, as that was really a secondary endpoint for us, we felt that it was possible to continue with the trial. Step 5, or the final step, is really, to review the data and plan ahead. And this is really critical to help you identify problems early on. So we would definitely recommend, and from our experience, reviewing the data and cleaning it as the trial proceeds is really important. Having regular oversight meetings so that you can identify and act upon issues, and really sitting down early to try and plan your research outputs, be they abstracts, publications, or talks. It's often not until you sit down with the data and try to see what the story may be that you identify gaps, which you can then readily make changes or amendments to your protocol to address those, and really, again, ensure the success and relevance of your study. And the final point I will say is it's always good to start thinking about what your next pragmatic clinical trial is. So finally, I just wanted to mention a couple of points on why we think, or I certainly feel, the REACT pragmatic clinical trial works. Firstly, this is a modality of research that is really accessible to everyone: trainees, fellows, early and later career physicians.
So the last speaker is Professor Sean Treweek, and he is Professor of Health Services Research in the Health Services Research Unit at the University of Aberdeen. His research interests are mainly in efficient and pragmatic trial designs, and he's leading the Trial Forge initiative that aims to be much more systematic about how we generate and use research evidence in making trial design, conduct, analysis, and reporting decisions. And tonight, Sean is going to talk, the title of his talk is Why Not Thinking About How We Do Our Trials Leads to Research Waste. I'm going to hand over to Sean.
Thank you. So hopefully oops, Daisy, straight away that's disappeared. Great. Okay, hopefully you can see that. Looks fine for me. And if it doesn't look, I'm sure the technical people will tell me. So I'm going to give a big picture overview of why not thinking about how we do our trials leads to research waste. And the first thing I'm going to do is talk about an old paper. So many of you will recognize the person pictured here. This is Doug Altman, a real giant of medical statistics who died a few years ago. 25 years ago, he wrote the paper that we're looking at right now, The Scandal of Poor Medical Research, where he talked about the problems that are routine in much medical research. And the only regret that Doug ever had about this paper was that he chose the word poor rather than bad. I wish it had been called The Scandal of Bad Medical Research. And we're talking about efficient trials today, and clearly bad medical research, bad trials are the ultimate in poor efficiency. In that article was this text. So this is the front page of a notebook, in fact, that I actually give to new research students as a aide memoir to what we ought to be about. We do not need endlessly more research. And I'm going to show you some examples of that in a moment. We don't need more and more and more. What we really do need is better research, and research done for the right reasons. And we've heard from all of our speakers so far about the sorts of things that make the right reasons, meaning that research is going to be meaningful to those individuals who are trying to make decisions about better healthcare. Now, this little task I'm going to demonstrate to you now is something everybody can do in the privacy of their own home. You can do this at any time. I've done it several times, and that is, let's look at the Cochrane Library. Let's go online, look at the Cochrane Library, and look at the reviews that are flagged under highlighted reviews. And I did this last week. I've done it before. Here are 5. I don't know anything about these reviews. They're just the ones that were highlighted when I looked at this last week. And the purpose of this exercise that you too could do if you wanted to is, let's look at each of these reviews and look at one part of it, and that is the risk of bias assessment for the studies, the trials that are included in these systematic reviews. So these are all big systematic reviews. They've searched the global literature and pulled out trials in quite diverse areas of research. So we have something at the top there looking at diet in preterm infants. We have a surgical one there, the second and right at the bottom. We have something about mechanically assisted walking for people or children with cerebral palsy. And the task is, let's look at those risk of bias assessments and see what they throw up. So that risk of bias assessment in these reviews for each of the trials is telling us something about, should we believe these results? Have they done things which suggest that there's real doubt as to whether we should believe these results, whatever those results are? And I'm going to break it down into essentially a couple of categories, which is good and bad. So good means no, there are no reasons to doubt the way they put together the studies, so that would be low risk of bias. And bad means high risk of bias. Well, there's plenty of reasons to be concerned. So I did this last week, and this is what I found for the first one. In that systematic review, 7 studies, 1 of them good, 3 of them bad, and the other ones are uncertain. So we're not quite sure. Right, all of them. So the things to look at, what we really want to see are numbers of good that look very similar to the total number of studies. And if you put your eye down there, what you'll see is actually there are quite a lot of studies, and there are very few that are good. In other words, there are very few that come out as being at low risk of bias. There are plenty of reasons to doubt them. And if you look at the number of studies and compare that to the number of bad or the number of good, what you quickly conclude is that there are entire bodies of evidence that have been collected, linked to a particular systematic review question, where that entire body, or almost the entire body, can be classified as rubbish. Highly inefficient, clearly. But just look at it. It doesn't matter when you do this. I've done it before. You do it next week, you'll get the same result. It doesn't matter. And this should deeply shock us. There are hundreds and hundreds and hundreds and hundreds of trials that come out of this sort of assessment as poor. That should bother us. So the billion-dollar question is, to use Doug's word, why is so much health research bad? And obviously I'm focusing here on trials. So Doug himself in 1994 actually gave a very good answer. There's a lot of research that is done because researchers feel compelled to do something, to do some research, and they do not have the skills to do it, and nobody stops them. And we therefore get a proliferation of research that answers nobody's questions. It's weak in many, many ways and represents gold-plated research waste. If you remember the last slide, there was a bit at the bottom for cerebral palsy where the entire body of evidence in systematic review was a high risk of bias. You just didn't have confidence in those results. Nobody stopped them doing those trials. And why is that? Why did they do it? That body of research, not just a single study, it's not just one bad apple, everything in that entire area. So what I want to do is give a few what I think are top tips. And actually, this is merely summarizing many of the points that we've already heard in this session already from our previous three speakers. So there's no rocket science here. This is summarizing, repackaging much of what we've already heard. So the first thing is do something that's important. Find some priority question. We've heard already about the importance of the question. There are organizations and groups that do this well. They have the resources, they have the skills, they go out there, they ask people who are the people who we are trying to support, and they say these are the important questions. Guidelines groups, for example, often highlight gaps. They've hoovered up research evidence for a particular clinical guideline and highlight gaps in their evidence. We need to know something about this because that makes it difficult for us to make recommendations. These are the sorts of things that we should be doing with our research. It's an important question, and it's a priority setting process that has been done well. So if these exist, well, these are a great place to do an important piece of work rather than scratching about one Sunday afternoon in your own home, thinking of something that might be quite neat. These give you a head start with your question. And of course, here in the UK, the National Institute of Health Research does a lot of this. That's what their commissioned research is. These are known important questions. Top way of avoiding wasting everybody's time. Know what's been done already. I think it was Sharon who mentioned this, systematic reviews. If you are working up any research protocol, particularly a trial, and if you have not talked about systematic review, you really need to look and start thinking about systematic reviews. What does that body of evidence say? That Cochrane summary where I was talking about good and bad studies, if you could go to any of those systematic reviews, there will be some very big pointers in there to how your study cannot be as bad as all of the previous studies that are in that body of evidence. Don't repeat what they did because they're rubbish, a lot of them. So you need to know what the systematic reviews are saying so that you can build on what we know and this time generate some evidence that is really, really useful. And funders, again, the NIHR in the UK, are increasingly thinking about, well, where's your systematic review evidence to back up the rationale for your research question? If it's not there, we should really be questioning, well, how do we know that this is not research waste? This one, design for users, definitely been mentioned. Who are you trying to help with your piece of research, and what do they need? Not what do you need or what do you think is neat or might be quite interesting, but what do they need? What are the endpoints that they need, the outcomes to answer their questions? Make sure you know what your intended users want and design your study around them so they don't have to throw it away later on as being completely irrelevant. So the users are right at the center. Process is important. Let's not think that we know how to do a fabulously efficient trial, even if the question is really spot on. So this person, Mike Clark, he came up with a term called SWOTs, studies within a trial. And it's a great way of evaluating trial process alternatives. So we could have one recruitment method versus another recruitment method. What's the best way of recruiting our particular trial population? Well, if we're not certain, let's do an evaluation. So that's what SWOTs are about. Ask, why are we doing this? I think it was Greg who said that. Why are we doing this? SWOTs enable us to start providing evidence for trial process decisions, how to recruit, how to retain, how to collect our data, how to present our data, those sorts of things. Think about process. That can avoid inefficiency. And this point has definitely been made. Research is a team exercise. This is not something which one individual is going to do alone, or even just a couple, where we all have our own expertise, but few of us are experts in everything. And members of the public and patients with particular conditions are absolutely experts in that. We need their help. They should be there from the very beginning and throughout. And find a statistician, find Greg, become friends, and involve them from start to finish, so that you don't, at the end, try and present your half-baked set of data to a statistician and ask him or her or they to magic up some great piece of work. It's too late, shot. So that statistician needs to be there from the very beginning. So these sorts of things, I think, take you to what Doug is trying to encourage us to do. To think about what makes our research better. We don't need more and more and more and more. We need better research. So we don't repeat what we saw in that Cochrane summary, and research done for the right reasons, thinking about our users, knowing that it's a priority question, knowing that systematic reviews are pointing us in that direction. And I'll leave that with you and hand back to Marion. Thank you.
Excellent. Thank you, Sean. Lots of food for thought there. So we've had 4 excellent talks that I'm sure have generated lots of questions for you, and I'm delighted to see there are lots of questions coming in the chat bar. So I think we're going to start the question and answer session now. I see there are themes developing here. So I'm going to start. So the first ones were obviously stimulated by Mark's talk at the start about oral consent. So there's a question here from Natalie Blenko, who's asking, do you ever run into difficulties using shorter consent forms with the ethics committees? So I think we'll start with Mark. Do you want to take that one first?
Thanks, Natalie, for the question. I think the inner surgeon in me is blessed with a belligerent personality and has been able to keep going at the REB about these. So if you're the sort of person who's going to give up at the first rejection, then pragmatic trials are not for you. Yes, we have faced multiple challenges with the ethics board. As I said in my talk, it is not a lack of consent, and what I often say at these meetings is a 20-page form that's been signed is not consent. It's just a 20-page form that's been signed. It means nothing. And the beauty of the integrated consent process is it's part of your normal conversation with patients. It should be that we're offering multiple different standards of care and accepting that we don't know what's best. Now, it's very difficult as physicians because we always think we know what's best. The reality is we often don't. And the REBs have been extremely receptive. And what I normally do is every time I submit a protocol, we send the New England Journal papers on oral consent processes with the application, because our REB has regular turnover of staff, and it feels like we're having the same conversation over and over again. So yes, it's a challenge, but don't give up.
Excellent. Thanks for that response, Mark. And so I have another question, probably straight back to you, Mark. And this is from Alison Fielding. So do you have any issues with patient dropout after the clinic when the patients go home and then they consult with family? Do you then find that the patients will drop out having already signed up to the trial in clinic? So again, over to you, Mark, for a response to that one.
Thanks, Alison. Another great question. So when I'm auditing somebody else's trial, if I find that there are no dropouts, it kind of raises a red flag to me that something's going wrong. So we actually published this as part of our consort diagram. We'll say how many people were randomized, how many agreed, and how many, what the acceptance rate of the randomization arm is. Dropout rates are actually incredibly low, I'm pleased to say, but yes, we collect it. And it's not that, again, that this is avoiding consent, it's having those honest conversations with patients, and they don't get randomized until they've made that decision. So if a patient needs to go home and discuss it with their family, absolutely fine.
Excellent, thanks for that response. So there's now a couple of questions about outcomes. So I think Greg will be in the firing line for these ones. And it's an interesting, there's a phenomenon in statistics called sample size samba, which is the dance between the clinician and the statistician. And the statistician says you need 2,000 patients, the clinician says I can only get away with 200. Please. And then there's this sort of slight dance around where we get to a common decision point between the two extremes. But the question probably for Greg is around from Mahim Qureshi. Do you have any advice on composite outcomes? Because clearly they may be more efficient and may help you with sample size calculations. But do you have any advice on that and on how to construct and then test a composite measure to inform that sample size calculation? So Greg, a response from you?
Sure. I mean, composite endpoints, they can be a very good thing, but they can also be problematic. So one of the big concerns about composite endpoints is if each endpoint measures slightly the direction of the outcomes are different. So you can have one which is wildly in favor of, say, the experimental arm, one which is moderately in favor of the control arm. When you combine them, you're going to get a neutral result and a negative result as a result. So one of the key things when you're talking about composite endpoints is you really have to understand or have to believe that the outcome is, or the direction of outcome is going to be the same for both outcomes that make up all the outcomes that make up the composite endpoint. You see that, I've seen that a number of times. I don't do cardiovascular, but I've seen it in cardiovascular all the time. Because the event rate is so small that they make a composite endpoint about 4 or 5 different outcomes, and unfortunately sometimes one goes in one direction and one goes the other, and it ends up being a neutral thing. So you have to really understand that. The other thing is, you really, just getting into the understanding of it, you really have to make sure that one outcome doesn't overwhelm the other one. So if say one outcome happens in 15% of patients, and you've got 2 other outcomes which happens in 1 or 2% of patients, there's not really much benefit in having a composite outcome in that case. You can really power it based on the single large outcome that, again, hopefully that's clinically important, but you have to really focus on that. So a lot of it just comes down to understanding what is really important and what's going to happen, the direction of the outcomes in favor of the standard or the experimental arm, making sure that they're consistent, and thinking it through. Again, at the end, one of the things you really have to do is think through at the end of the study, if I get a composite endpoint, is that important? Or is it more important if one of the outcomes was more really what's going to be the driver behind everything? Should I focus on that one outcome as opposed to having a composite one?
Thanks, excellent advice there. And just keeping on the theme of outcomes, I think we'll probably ask Sharon this one. So Natalie Blenko has said, given the tensions between different possible primary outcomes, how do you optimally select between primary outcomes? Which one do you use in the end, and how do you reach that decision? Sharon, do you want to comment?
Yeah, thanks so much for the question. It's a great question. And really our approach in the pragmatic trial program is to really involve the people who we're hoping this research will impact, which is the physicians and the patients. So, from our, data review, we might have certain endpoints or outcomes that we think are interesting or relevant to look at. And then usually our practice, is to go to the physicians and also ask patients what is meaningful to them, what is going to change their mind mind or their approach about how things are done. And, you know, obviously taking into consideration whether it's something sort of feasible that the readout would be relatively, you know, quick or possible to do, we would usually focus on that. So that's our usual approach to picking an endpoint, you know, seeing all the relevant ones and putting it to the patients and physicians who are really going to be using your data.
Super answer,
Marion.
Sharon. Yes.
Marion, if would it possible just add something it follows in beautifully from what Sharon has just said, if that's okay. So in exactly the same way you've just described, Sharon, we were looking at a bunch of trials published, I think they're in the New England Journal of the Lancet, and we got all of the outcomes that they described, so the primaries and secondaries, mixed them up, and then presented them to a panel of healthcare professionals and patients or patient representatives to ask them to rank them. So we didn't know what was the primary, what was the secondary. We asked them, if you were making a decision about this treatment, what was the rank you'd put them in? And interestingly, there were 22 trials involved, and in only 6 of them was the primary the number one outcome, in the sense that the trial team had not made the right choice in most cases. The primary was highly ranked, like in the top 5 in most of those trials. I can't remember what the number was, but, you know, it appeared in the top 5. But there were a couple out of that 22 where the primary was nowhere to be seen. So I think it's very important to discuss with your future users your outcomes, because you can mess up royally. You know, they don't care at all. It's possible, and we clearly want to avoid that situation.
Excellent. Thank you for that, Sean. And I suppose just building on that theme, I was going to ask Sharon and probably you, Sean, as well, where does this sit with core outcome sets? So there have been a number of core outcome sets developed for certain disease areas where that piece of work has been done, whereby the different stakeholders have tried to address the questions and the outcomes that are important. In what situation should one use the core outcome set as the first port of call? And do they exist in the trials that you've been involved in, Sharon, or is it very much the clinician that leads on this? So Sharon, I'll ask you first.
Yeah, I think it's more so the clinician and the patients that lead. You know, the core outcomes, you know, are certainly important, but sometimes that might already have been addressed in a certain clinical question, and it might not be what's, you know, stopping physicians or patients from making a decision about treatment. So, you know, they are there, and they will often be included in what we present to patients and physicians as possible outcomes of interest. And if they, you know, reiterate or select them as of interest to them, then definitely we would look at them. But really, we're more driven by, you know, what is helping physicians in the clinic and what is guiding patients.
Excellent. Thanks for that, Sharon. So maybe actually I'll just pick up another question, and I'm going to go to you, Sean, this time. So this is from Kristen Hoffman, and she's been commenting on your talk and saying, what would you recommend as the most efficient and most important change that could be implemented to increase the quality of health research? That's a big question, Sean. You're up for it.
Find a methodologist and a statistician, and possibly the same person. So in my experience, I mean, I'm a methodologist. This is my conflict of interest, but not a statistician. But if you look at many research proposals, when you're on, say, a grant funding committee with colleagues looking at a piece of work, it's possible often quite quickly to find really substantial holes in it. And you look across the team and you find that there are no individuals within that team who have expertise in method or stats. There's lots of expertise in other areas, but not those areas. And that, I think, if there was much more acceptance that research method is an expertise, and if you do not possess it yourself, you need to go out and find it, because without it, you're in danger of doing more research that nobody wants. There's an opposite of what Doug Altman was saying. So statisticians, methods people, find them, embrace them, and involve them in all of your work so that we have our full team of expertise.
Super, thanks, Sean, and I'll pay you later. Being a statistician, I love the fact that you're finding friends for me, so that's great. So another question, I'm going to turn back to Mark again for this one. So Samir Patak had been, like the quote, if you ask for more, you will get less. And certainly in the UK and maybe in Canada too, there's a lot of time spent filling in screening logs. So you log every possible patient, whether they're eligible or not, whether they ended up getting randomized or not. And it takes an awful lot of time and detail and commitment. So the question would be, what are your views on the collecting of detailed screening logs, and are they important? So Mark, do you want to come in and give an opinion on this?
Yeah, absolutely. So let me just move the camera. So you see a pile of boxes there that are much smaller than they were last week. This is data that has to be kept for 25 years as per our ethics. And what a horrendous waste of time and money. And so we collect superfluous data all the time. And one of our rules of thumb when developing our React protocols is with every iteration, the protocol must get shorter. Very simple rule to stop having too many cooks in the kitchen and everybody adding their thing. Logs are fascinating. So one of our institutional policies has been that every potential physician that might see a patient with that condition should be on these logs. We've changed that. We asked the docs before we start a study, are you or are you not going to approach patients for the study? And if there is no reply, we don't put them on the logs. This irritates people at other centers who are still stuck in previous models. But I think the methodological point of view is we collect far too much data. I was dreading, I thought you might ask me about basic science. Because if ever there is an example of something that really hasn't added much to clinical practice, yet you're unlikely to get oh God, Greg won't speak to me now. And, add a bit of basic science in there and it'll score you more points. And I know that I work in a lab. It is a lot easier to write a laboratory grant than a clinical grant because as clinicians, we live in shades of gray. We're used to the fact that nothing is precise, no endpoint accurate apart from death. So I'm going off topic here, but these are huge issues that I find very interesting. So that's the grants gamesmanship, another session for another day.
Mm-hmm. Super, excellent.
Can I just add, I just wanted to add in a couple comments because we did a study in our local clinical trials group a number of years ago, and we found something about 30% of the data that's collected is never used at all. Particularly if you look at baseline comorbidities, baseline medical history, these are things that are routinely collected and just rarely ever looked at after the fact. So these are areas where you can really shorten a protocol, any protocol. If you're not really interested in it, you don't need to have to collect it. The labs is another area. And one of the other things that we looked at is, this was in Canada a number of years ago, but for every data point collected, we estimated it was about $50, 50 Canadian dollars, about 25, 30 British pounds per data collected point. And that's per patient. So if you have 1,000 patients, that's, you just multiply that data point by 1,000 and again, it just explodes exponentially. So you can see how it increases cost, increases workload, and there's not a lot of benefit if you're never going to use it. So really think through even the CRFs, what data points do I want to collect at the beginning of the trial? Superb.
And so we're getting near the end of our session. So I'm going to finish off with a really excellent question from Emily Kirkham. And, her comment is, do you think too much emphasis is made on getting your work published for the sake of the CV fodder, rather than getting the trial design right so that it's answering the right question? So this one I'm going to ask Sean to come in first. I'll probably ask you all for your views on it.
Yes, I think so. Yes, I think it's easy for me because I'm now old to say yes, and the problem is that the academic career progression process has perverse incentives for publishing nonsense, and as opposed to spending plenty of time doing that really great design and getting a really quality piece of work, and then spending plenty of time doing other forms of dissemination of that single academic paper. So yes, I think they have a big job on changing it, and it is much easier for people like me to say, well, let's just not bother publishing, than it is for early career researchers, because of the way academic career progression is set up. It's bad.
Yeah, and I'm going to ask Mark as well from the surgeon's perspective on this.
I'd hate to be thought of as a surgeon. I said I have surgeons.
Oh, sorry.
I have.
I thought you were getting your inner surgeon on at the last one.
No, my personality disorder is very surgical, and it's sort of my belligerent, the sort of person that wants to get things done, and those are magnificent traits in a surgeon. Absolutely. Research for the sake of doing research is extremely widespread. Funding, and the UK is the same as Canada. It's very, very difficult to get grants. And there's a disincentive for forming teams, big teams, because people are competing with each other. And we know that on the grant panels, same old tends to get the grant money. It's perverse incentives. I agree, and I don't know what we're going to do about it. And I agree. Those of us who are on the nearer to death end of the life spectrum have less to be concerned about. But I do. We publish a load of trash.
Okay, so I think it's probably time to wrap up the session. So my learning points that I've taken from this is that you need to think about your trial design from each point. You need to think clearly about what outcomes matter, which ones you're going to choose, how you're going to measure it, how big your sample size needs to be to ensure you get that. Keep it simple. Minimize all the surrounding trash, as we've been heard. Keep the surrounding excess paperwork simple. Keep it fundamentally focused on the trial question at hand. And if we do that, we will fulfill Doug Altman's aim to do less research, but better research. And I think that's an excellent point on which to wrap up the session. So I just want to thank all our speakers for an excellent set of presentations, an excellent set of questions and answers. And I want to thank everybody who's participated in the webinar. You've had great questions coming in, keeping the chat flowing. And I hope you've learned a lot from this. And I'm sure our NRCM colleagues will be inviting you very much to sign up to the future webinars that are coming on stream over the next week or so. So I'm hoping to see you there over the next couple of weeks at the next webinar. So thank you very much for your contribution tonight.